Good morning. Good morning. Good morning. Good morning. Welcome. It's Friday. It's the weekend's going to be here any day now. You did it. You got to the end of the week. And it's supposed to be a good weekend. Get outside, do something. It's supposed to be snowy. Today's going to be sunny and a little bit cooler. And I gotta say, as a backyard rink worker, I'm really loving this winter so far. Thank you for finding these words. I'm going to take a second and clear this work, but you can do these again at your leisure as you're going through the PDFs later on. Clear all drawings. And we're going to go ahead. And we're going to, before we get to where we're going today, it's February, my calendar tells me, which seems strange, but it's February. So it's Black History Month. And that means we have an opportunity to kind of share stories that otherwise have not been shared for deserving scholars that overcame societal hurdles to become who they were. And today we're going to talk about an intersectional hurdle exceeder, Dr. Joan Merle Owens. You can read more about Dr. Joan Merle Owens if you follow that QR code later on, because Dr. Jessica Ware, who is a Canadian at the American, she's the curator, the head curator of invertebrate zoology at the American Natural History Museum in New York, like the one in Friends and Night at the Museum, that kind of thing. And she's doing this hashtag 28 Days of Black STEM History. And Dr. Merle Owens was one of those. And Dr. Merle Owens is a scholar, was a scholar of Nidaria. And you are scholars of Nidaria, so we need to understand the community that you are now part of. She was born in 1933 and grew up in a segregated South. And she entered college in 1950, as did most of her colleagues and friends. But she was told that because she was black and a woman, that she couldn't do this. So she majored in fine art, and she received an MA in guidance counseling. And she pioneered a teaching program, a national teaching program called Upward Bound, that was meant to mentor equity-deserving students and youth with people in colleges and universities across the country. And she had a 15-year career in teaching until she was 37, and she decided this is not what I want to do. And so she re-enrolled in college in a program that had biology and geology. She did a second degree because that's what she had to do. And after the second degree, she knew that she wanted to go to the ocean, and she wanted to learn to scuba and to explore the ocean, this place that had fascinated her since she was a child. But she learned at that point that she was a sickle cell anemia carrier, and that it was too dangerous for her to dive. So, new hurdle, new leaping of that hurdle. She embarked on a plan to study existing collections of marine corals that existed in the Smithsonian. Some of these are taxa and specimens that have been collected 100 years before. She was awarded her PhD at 51, which I think is a young person, because I'm 53, but this is again someone pushing through societal barriers. And she exceeded even more because she became a professor at Howard University and was a professor there until 1995, and won awards across the country for her passion for teaching and research. And she described new species of coral and new genus of coral and of button corals. And these were kind of very small. They're about the size like a loony, between a loony and a quarter. And they live in isolated fashion. They don't live colonially like the corals that you know about. So, tip of the hat to Dr. Joan. And I see in the chat, yeah, go and check out the picture that Sherry posted. This is another opportunity, though, for me to call attention to this. Maybe you've seen this already. I'm going to keep the slide up for a second, but you can scan either of these QR codes to learn about this new program, relatively new, 3 years old, FREED, Field Research in Ecology and Evolution Diversified. So if you are an indigenous, black, or racial minority as an undergraduate student at the University of Guelph, so an equity-deserving group, this is an opportunity. This is a specific field course to give you opportunity to get outdoors and to gain experience. And these pictures are from years past when it was in Algonquin. This particular year, it's going to be more accessible and local down the highway at the Rare Research Reserve near Kitchener. And there's going to be applications already open. That's the QR code on the left. And then there's an info session that'll be in a hybrid fashion in the Science Complex 1511 or in one of Zoom, or let's hope it's Zoom. And God, I hope it's not in Teams, but you can persevere if it is. And then if you have questions, you can send Martha or Alexandria a question, and they will give you an answer. So let's begin. Previously, on Zoology 2700, we were talking about the Lophophorata, these three phyla, the brachiopods, the bryozoans, and the phoronids, or the muppets, that possess this organ, this coelom-enabled ring of tentacles that surrounds a mouth, that tend to live sedentary lives as adults and are part of the holoplankton as juveniles. We talked about bryozoan, colonial, larval stages that are pretty cool. They have the siphonaut form. They have contributed mass amounts to our own species in our capacity to combat HIV, AIDS, cancer, leukemia, Alzheimer's. We talk about phoronids and their respiratory pigments and why they might need one, which is generally a trait that's associated with doing like active things, but they live a really sedentary life. And then we talked about brachiopods and bryozoans that have existed since the Cambrian, like into the Paleozoic, and their peak of diversity was actually in the Paleozoic, not in the Cenozoic where we are now. And those Lophophoriates were, of course, in chapter 17. Today, things are going to get wormy. And this is the true worms. We're going to be here for a while. This is the most diverse taxa that we've talked about, and it exemplifies a strategy in metazoan development and growth called metameric segmentation. So we're going to talk about that, but really these are the true worms. And some of them are, as the sand striker there on the left, are terrifying. Some of them are gorgeous. They're all gorgeous. We'll talk about the synapomorphies that characterize them. We'll talk about one particular lineage, Turbiani polyphyletic lineage that I was taught called the polychaetes, that we more contemporarily call the Arantians. These are active ones, generally, like this one that was paddling by on the right-hand side. We'll talk about nutrition, movement, circulation and respiratory pigments, excretion and reproduction, and all of that's going to be today. And it's going to fit these learning outcomes that you are becoming familiar with. Sitting under the big context of being able to evaluate evidence based on prior experience when exposed to new things. So to appraise, synthesize, and interpret examples of all of those kind of categories or statements and new areas, the synapomorphies and the ecology and evolution of this group. Let's begin. The annelids here. So we're still in the spiralians. We're in the protostomes. So we're in the Dun phylogeny. We're in the protostomes, and we're in the spiralians. The spiralians we're going to be here for a few more weeks because they have the platyhelminths, where we've been already, parasitic and free living. The rotifers also kind of groups around 3,000, fewer than 5,000 species. But the spiralians contain the annelids and the mollusks, which are going to be some of the most diverse groups that we come across. And most fascinating. Often when I say we're going to be talking about worms, the mental image, the little kind of bubble of thought that I see appearing above your head is of an earthworm. This is an annelid. Much of the diversity we're going to talk about is marine or parasitic, i.e. the polychaetes or the leeches. We're going to talk about earthworms. You're going to see earthworms in labs, but because you're dealing with them in lab, we're going to spend less time with them here. So some terrestrial worms, but we're going to celebrate the diversity of form. I'll give you a kind of a shout out to next week when some of the, you have fewer phyla to remember than I did back in the 20th century when we could barely communicate with each other. Things were technologically so poor, when the phone only existed on the wall in your parents' house, not in your pocket. And at that point, there were at least 3 other phyla that have now been put into the annelids. And so we'll talk about those because they're really divergent. And so there's a whole bunch of forms that we're going to talk about. This is kind of exciting to me. But before we get there, let's talk about a big picture metazoan appearance of a strategy. And that's these metameres. So annelids have 3 body regions that you're looking at here. The pygidium, the peristomium, and the prostomium. Prostomium is like the head. That's if there's eyes or ocelli, that's where they are. Peristomium, kind of the neck. And the pygidium, the tail at the back end. And in between, there are these replicated body segments called metameres. Segments, segmentation. Now, you've seen kind of a replicated form when we talked about the cestodes. You remember those strobilglottids? Those kind of the bag of eggs that break off at the end and go out in the feces? It's early in the morning and already Smith is onto the poop. Now, the growth zone for this strategy is different. Remember with the strobilating proglottids, the oldest area was just beneath the scolex. Right, and sorry, the oldest area was the farthest from that point because they produced here and then they break off down there. So these are the oldest. Whereas here, this growth zone sends segments that way, anteriorly. So the head, the prostomium, is kind of the oldest thing, and that's because this growth zone at the anterior end of the pygidium has these teloblast cells that form each segment. And these teloblast cells are large cells that are a characteristic of this group that divide to form many smaller cells that then proliferate and differentiate and become the metameric segments. So metameric segmentation. Each segment, each metamere, contains all of the elements of any of the other segments. So there's a pair of mesodermal somites with coelomic spaces. There's a pair of nephridia. We'll talk about that for excretion. There's a pair of coelomoducts that allow the passage of excretory products and reproductive things. There's a pair of ganglia, and there's a pair of appendages. So here's a really simplified. So you see the gut proceeding through all of these things, but you've got the septum right here. That's this dividing wall. And the dividing line, those are the edges. So the little bulges there, those are the metameric segments that would have each of those, a pair of nephridia, a pair of coelomoducts, and a pair of ganglia, and a pair of appendages. And like I've said, this is not like the cestodes. So these are proglottids, and there's the scolex, the head up here under my mouse, and then older things, older eggs breaking off. So this is, metamerism is not like the strobila of the cestode. Okay, and so the oldest ones, you see the oldest ones are completely the opposite end of the body. That's, I don't know, maybe one way of kind of orienting yourself if you're comparing and contrasting. This is an older line drawing from the text that preceded this text that has that you can kind of go through, but it breaks apart one of the metameric segments to say, yep, there's a nephridium, there's a coelomoduct, there's a nephridopore, and we've got replicated elements of all of these things going through the length of a truly metameric organism. There it is again in red on the bottom, but we've got segmentation. That's the cestode, where the segments are derived from the ectoderm, so they kind of grow on the outside in, whereas in the metameric segmentation, they're going from the inside out. So lots of advantages to this kind of a strategy. You can build a large organism from a simple plan, and you know this because probably you grew up playing with Lego blocks, right? I did. If they existed when I was young, perhaps they existed when you were younger as well. Those are metameric segments, right? You build a large thing from a simple plan. And it means that you have the capacity to effectively service these major systems like excretion and reproduction and muscle systems, even circulation in major ways. You can have flexibility. You can have complex movement. Loss of parts is generally not fatal. And the next thing we're going to talk about, you have an opportunity to specialize. Because just like Lego, you could build the same series of bricks and you could make a very simple metamerically segmented worm marching from left to right. But there's, within the metamerically segmented organisms, there's a process called serial homology. So serial homology means that you have the specialization of appendages and other structures for different functions. So the difference between, for instance, antenna and legs, mouth parts and legs, this is specialization of a lot of these appendages that would have been on individual segments. So even though they have the same embryonic origin, they are utilized in a very different way. And then the ultimate, at least in an evolutionary and diversity sense for animals, is that you can then have something called tagmatization. So a tagmata is when metameres, those segments, are grouped together, so specialized serial homology, so they're structurally different. And you know this already because you've been able to draw an ant since you were in kindergarten. And those three circles that I just sketched, those are the tagmata: the head, the abdomen, and the thorax. Look at that. Look at that and reflect on how much better your lab notebook is than that. Okay. Annelids. So sipunculans and echurians, those were worms that were different phyla when I took this course. The polychaetes and the sedentarians, so Erantians, mobile marine worms, sedentarians, mobile and terrestrial and freshwater, slow-moving worms, and then the hydrodinomorphans, which are leeches. And we're going to get to, over the next several classes, talking about specialization and some amazing stories about the morphology and evolution of all of these different lineages. There's a lot of species, and they're in the 14th chapter. And you've seen some of them already, because if you've been watching some amazing ones, this one looks like Cousin It from the Addams Family, but this was filmed, 2 mornings? What is the day? The 7th? So Wednesday morning. This was an annelid that they found, an Aphroditidae, so a sea mouse that has these long bristles. So the individual segments are and the parapodia that are moving it are hard to see. They're underneath all those long bristles. But this was underneath about 1,100 meters underneath the Antarctic, surface of the Antarctic Ocean. Again, don't memorize this. Don't try and draw it. This is the kind of phylogeny, a phylogenetic hypothesis for the major lineages or some of the major lineages of the annelids. This is one that I've kind of simplified with some colors here, and we're going to come back to it again and again to remember where we are and what groups we're talking about. So there are two major clades, two major groups, the Erantians and the sedentarians. As the names maybe make you think, if you think about, so an errant thing is a thing that is mobile and moving. Doug and Lily leave class. So things that are Erantian are like lots of the mobile marine worms, formerly known as polychaetes. And then the sedentaria, so they have parapodia and nuchal organs, so sometimes very sensory. We'll talk about those as some of the synapomorphies. The sedentarians include things like some of your earthworms, that kind of thing. So clitellum, cocoons, no appendages, so no parapodia. Let's talk about one of those. So you saw the bristles on the Cousin It, the sea mouse scurrying across the bottom of the Antarctic Ocean. That's actually, those bristles are one of the synapomorphies of the annelids, and that's chaetae. So these are cuticular structures that you're looking at here on a very chaetae reduced. This is like using me, this kind of image, this SEM, is like using me to demonstrate hair. I mean, why not show, I should be showing the sea mouse at this point, but you can zoom in and take a look at it. Okay, so there's 4 simple bundles of chaetae per segment. I'm going to mute for a second and pardon me, I hope that didn't make it through. And they form the service of traction. And then other tasks. And I would imagine that the sea mouse, it's not so much traction. But in the paddling parapod that you saw at the start, oh, my apologies. Oh. Let's try that again. So, In the sea mouse, a lot of those chaetae weren't being used for traction. Some probably were, and in the paddling parapod that you saw in one of the earliest slides that had the sand striker, in that pink kind of paddle forming segment, those were the parapods, definitely had chaetae on them. So this cuticular structure that is different than setae. Setae and chaetae are different things because of the type of chitin that they're comprised of. So on the left-hand side of these two diagrams, you can see this is in an annelid. This is the thing we're talking about right now. So it's this little kind of cuticular outgrowth made of beta chitin. Beta chitin, when you cut it up in cross-section, it looks like this. So tiny, like a fiber optic cable. So lots of different small elements. Adjacent parallel with the same polarity that's very tough and very flexible. And so it's very good to be able to stick into a substrate to help you move forward. Now, for the arthropods, when we get to them in the second part of this term, the arthropods have a really similar feature, but it's called setae. Now, setae is made of a different kind of chitin, which is just a different kind of arrangement. So they have the opposite polarity, where parallel molecules, adjacent and parallel molecules, have an opposite polarity with a pulpy cavity. That's what you're looking at in the cross-section here. And it's harder and stiffer, so not as flexible. So big difference, beta, flexible, alpha, stiff. And so chaetae and setae. So these bundles of sclerotinized chitin. Now, Caramel is going to remind me to look down at the chat because I see there's a whole bunch of things. No, so so it's a continuum, right? These are not binary things. Aurantians move more. Sedentarians can move, but move less. And in between, there is a gradation of you'll see some really fast, there's Tomopteris is a polychaete that is like a crazy paddling thing. And then you'll see some of the polychaetes, so an Aurantian, that actually lives in a tube, and its movement is restricted kind of up and down inside that tube to try and capture prey. So it's still movement, but it's not like paddling around the world. And then you can see lots of other things. You've probably seen leeches moving across almost in a geometric, a caterpillar-like motion moving across. So a continuum, not a binary. This happens a lot in biology. That's right. I'm looking for the, so I'm looking around. So sedentary, so continuum. A sentient duster, that's awesome. Why so many worms? Amazing. Oh, and actually we'll get to that one. I'm gonna talk about that one, the bobbit worm. That's amazing. Okay, I think I caught everything. If I haven't, post it again, and I will see it. Thank you, Caramel. So more of the synapomorphies before we get to drill into some of the stories. So this parapod, what am I talking about? Well, each segment has an appendage that's called the parapod. So paired muscular bristle-bearing appendages used in locomotion and sensing and respiration. So these, a pair of them on this is a bilaterally symmetrical thing, as you can see by that diagram, right? So on left and right side, they have, and in its most simple sense, it's a paddle. Think of this as like the dragon boat where everyone's kind of paddling on the outside. They're not cilia in this case, but they're a paddle on the outside. Except, so that's locomotion. Some Aurantians use those parapods as an external surface for respiration. Some polychaetes, Aurantians, use those parapods as a way to sense light. They see with their feet. And they're formed, these things, these little, and there's 1, 2, 3, all the way down the length of the body, an evagination of the body wall. So it's a body wall that kind of, so it kind of folds out, and then you've got your parapod. Now another one is the nuchal organ. The nuchal organ, so moving down here, is right up here. So there's the parapodium near the front. And it's a little pit. And what you'll find, especially if you come back and share 3700's enthusiasms with me and Sherry or Shayla, is that these pits are a great way to collect information, whether it's chemoreception, photoreception. So a ciliated pit or groove that can detect photoreception is light detection. Chemoreception is like tasting the water, moving towards food, moving away from light, often away. So like a negative phototaxis. Or in this case, one we're moving away from shade because shade could be a predator. That's accomplished by these little nuchal organs. Now they have something called pedal ganglia or parapodal ganglia. So there's the nuchal organ and the parapodal ganglia. So these are facilitating the coordination of these paddles, because you don't want to have like some kind of alligator walking, that kind of offset, that reptile movement. That's great with only 4 appendages. If you've got zillions and zillions, it's harder to coordinate. And in fact, what you're going to see is that there's a power in a recovery stroke, just as there was with those metachronal waves of the cilia. So there's a high degree of coordination that has to happen. This is one of the most complicated nervous systems in any of the taxa that we've come across so far. They are here. How do I further support that statement? Oh, I want to go back and say there is a nuchal organ. This is a little pit sticking off the front end of an errant polychaete. So into that pit, there's lots of different cilia, and I'm making like a Snape kind of potions stirring. That's where the chemoreception or the photoreception takes place. Photoreception, Smith. What are you talking about? Well, because there is a degree of cephalization in the annelids. There's a brain, right? You could have zombie worms. There is an anterior dorsal ganglia that is large and concentrated. So there is a brain with large ventral pairs of nerve cords going down, okay? They also have prostomial ocelli, which are, I think you will agree, looking at them in an SEM and then a color photograph here, pretty much eyes. Okay? They have, in cross-section, they have a lens for concentrating the light. They have a sensory pigment and sensory cells at the back end of this vitreous body and then connected to an optic nerve. This is to all intents and purposes, this is a simple eye that certainly lets them see light intensity and direction. Whether or not it allows the formation of images is not entirely clear, and my guess is it will be because of the diversity of strategies in the annelids. My guess, if you want to go out and prove me wrong or support this prediction, my prediction is that in some lineages it will be, but not all. Some lineages are much better, they have much more endowed nervous systems. Case in point, Myxicola infundibulum. That little arrow in there is pointing to the largest nerve fiber that's known in the animal kingdom. In some specimens, it reaches a 1.5 millimeter diameter. So that's bigger than some of the individual organisms that we were talking about on Wednesday. Like, this is bigger than a ketognath. A ketognath could use this nerve fiber as a long tube to run its the ketognath reboot of severance. This could be the long tunnels that they move around. Okay, and that's specialized, long, thick nervous system, long, thick nervous fiber. This is about rapid transmission of impulses. Large diameter, low resistance, rapid conduction. And the conduction velocity can be as fast as ours. Again, a current that moves through the river of this course is the emphasis on the lack of how special we are. We are one of millions. And I love many of the members of this species, but it's not because we're so much better. It is not at all because we're better than any of the other ones. We're really bad at doing a whole bunch of things. In this case, the fast response has to do with escape in the Myxicola infundibulum. So then we've got the pedal. So I mentioned the pedal ganglia. Here we see it here. So this is controlling. You can see the nerve is heading out. And then a ganglia is basically just like a swelling, a concentration of that nervous system. And there we have it here within the ganglia, and that would be replicated down the length through the metameric segments of the body. We've got an eye there. We've got the cerebral ganglia, the brain. So lots of synapomorphies in the polychaetes that have to do with moving. Okay? So there's the pygidial cirri, which is what I called the tail in that long when we first talked about the segments and how they're formed from the growth zone, just anterior to the pygidium. Right? So the pygidial cirrus, those little tufts at the end, those are sensory appendages. So in the polychaetes, there's 3 major different lifestyles. You can burrow, you can crawl, and you can swim. So I ask you, put it in the chat. Where would you expect, which of these 3 groups would you expect to see modified prostomial ocelli? to see the eyes. Ooh, we've got a 1-to-1. Only 2 guesses so far. 1 for swimming, 1 for burrowing, 2 for swimming. I don't have a good... Oh, now for crawlers. And we've got 2 for swimmers, 1 for crawlers, 3 for swimmers, 1 for crawlers, 1 for burrowers. I can... Low risk, high reward. Yeah. Yes, I would agree with that. And I think you're saying there, Marlo, that's your reasoning behind choosing swimmers. So yes, or essentially, but remember, we're not talking about binary things. We're not even talking about 3 different silos, because there's a continuum, but these are concentrations. And this is not phylogenetic. This is like, so different lineages would have swimming taxa within them. Different lineages would have crawling taxa. Different lineages would have burrowing. But we have, the more active you are, the more likely that you are to see modified prostomial ocelli. So often in the swimming. Muscles. We've talked a lot about hydrostatic skeletons. We're going to keep talking about it here because this is really all about it. We've got circular muscles and longitudinal muscles and a coelom. So again, with my analogy of the water balloon, that you've got muscles and muscles that can help change the position. Now we've got more things than just an inching kind of appendage-free body, because we have the parapods. So we have different muscles now. We have these oblique muscles coming off of the sides, all right? And those head into, this is a parapod, this, I guess, wing-like thing, but you can see it has some bristles on the side. So when you see different shapes along the paddle with different bristles or spines, this is almost certainly from a taxon that uses a multi-use parapod, potentially for respiration as well as for locomotion. So here we go. The body wall of this hydrostatic skeleton is all about, it has a fibrous cuticle. So this reinforces and prevents bulging. But the collagen, you remember way back when we talked about collagen, David Attenborough taught us that it was like the most important protein in all of the animal world. The collagen fibers here are in a cross-helical arrangement. Okay, so this is what, like, orthogonal is at right angles, right? So that would be at rest, it looks fine, you've got your little resting pickle, it can shorten and it can lengthen, but if you bend the pickle, bad, because kinks, and a kinky pickle is not what you want if you have an orthogonally arranged collagen. If you have crossed helical, so slightly angled, that means you can shorten, you can lengthen, and you can very smoothly bend without kinking. And imagine, so the kink, what's wrong with the kink? The kink in this case is if you have the parapods, respiratory surfaces, coordinated legs, that kind of thing, that get jammed on the inside. That would be a problem. So the collagen is arranged in a crossed helical form. Many of them also make tubes. So in addition to that body wall full of collagen, they'll secrete tubes made out of protein, mucus, calcium carbonate, like many of the other tube building things or skeleton building things that we've run into so far. It's kind of what is in the environment around. So here we go. We're going to talk about feeding in a second, but Caramel's reminding me, oh yes, good. What are the muscles made out of? Same things as ours, right? They're composed of a lot of the same kind of, you would be able to identify an annelid muscle. Oop, sorry, Caramel. Swimmers, and let me grow up so. Oh, and again, I know what you were talking about, low risk, high reward. You were talking about making guesses here. This is where to guess because there's no risk, lots of reward. Like, make a claim. This is it. Participate in that way. I think that might have been what you're talking about. So well done. How are they different from eyes we're more familiar with? If they're what are they capable of? Seeing color? Oh my gosh, color. So a whole bunch of sight. We're not going to be able to talk as much as I would like to. The emphasis in 2700 are stories across the diversity of life. In 3700, we start talking about strategies more. And so there's multiple lectures on photosensitivity, photoreception, chemoreception, that kind of thing across the diversity of life. But in short, color, how we perceive color, is crazy ridiculously monochromatic. There are colors that dragonflies see because of the number of options that they have that we can't even imagine. And so it's not actually a simple question to answer as to what kinds of color do this polychaete see. It's an easier question. It's a lower hanging fruit to say, is it light receiving, yes or no? And so shadows and intensity. And there's a whole bunch of information in that alone. And then color, or different wavelengths containing different bits of information, that gets you into a whole other packets. Suffice to say, tying it to what I said earlier, how we're not good at a lot of things, I said earlier that we have a bias and that we see the world. We actually see a ridiculously small slice of the world that can be seen. And there are many other taxa that seeing wavelengths that we can't see. Google later on, we'll get to this when we get to arthropods, what a flower looks like to a bee. There's information there that we are unknowing, at best case scenario, unknowing and illiterate about. Let's unpack some. Yeah. Okay. This is good. We're missing so many colors. So rods and cones-ish structure. Those are kind of vertebrate terms that we can apply in an invertebrate world, and that we will in this course a little bit to come. How do they eat? Swimming, crawling, and burrowing? Well, if you're a deposit feeder, and this is maybe most of the biomass of the ocean ends up being deposit feeders, where they directly ingest sediment like this, or they indirectly, like this, with these gills and tentacles, collect and retrieve objects from the surface, pull them back into a mouth, and then retreat in this burrow and keep their gills up at the top where the water is more oxygenated. But they can also be predators. In addition to being suspension and deposit feeders, indirect and direct, they can be predators. So you have some business ends of tube-dwelling Arantians that have jaws, very business-meaning jaws that are tipped up with poison glands, okay, that are meant to grab onto a prey item, inject it with that poison or the venom, and so that it can then retract it into its burrow, into its tube, to consume it without damaging itself or its home. So the everted pharynx is used as essentially as a grasping tube. And this is, again, the orthogonal muscles, the antagonistic longitudinal circle that can squeeze circular to evert and then longitudinal fire to retract. So you can see a little bit of this. They can be active predators, as the one on the left, or more passive predators. So the cebalid or the duster worm, this is not actually what it might look like. It kind of looks to me like a cnidarian, but in fact, this is a bilaterally symmetrical cebalid worm that makes a membranous tube to capture particles with mucus. Don't go to a potluck with a cebalid worm. Probably also don't go to a potluck at this guy's house. This is a sand striker that you just saw eat a octopus. So sand striker worms live in mucus-lined burrows all around the world, all over the world. This particular one we're looking at, I believe, is from Indonesia. In rocks or sediments, and their jaws have sharp, sharp teeth. You can see when that video kind of loops back to the start again, all the different teeth on the jaws. They attack, they have no eyes. They have light sensitivity and triggers. So there, those hairs were reaching up, those antennae were reaching up to detect motion. And when something like this fish gill or this fish fin brushes by it, bam, they slam shut and retract. And this is a very bad day for that fish because things get very dark very fast. So two eyes, light receiving areas that lure and detect prey. They think that's also what that is doing. So during the day, they lie buried in the loose seabed waiting for a stimulus to trigger one of those things, to lure in and also to lure in the fish. But then at night, they actively protrude out. So in the day, those jaws, that trap jaw, will be right flush with the surface, kind of what we're seeing, right? But at night, they'll extend up and into the water column. Got about a minute here where we're going to watch
with multiple mouth parts displayed. Bone-hard grappling hooks, poised to pierce and immobilize prey. But the real nightmare remains unseen. Coiled, pent-up aggression as long as an arm. And some specimens grow to twice the length of a man.
Of a person.
As night falls, many sand strikers emerge to feast.
There it is, above the surface.
It's a bad neighborhood to hang out in.
It's a bad neighborhood to hang out in, indeed.
But fish are fast, and the sand strikers are
There's the
feeding
antenna.
blind. They don't have eyes or a brain. They react only if one of their antennae is triggered.
So no to both of those
if
things.
a shadow lingers.
Oh, that's a great shot of the jaws being retracted.
Patience.
Tip of the hat to this fish entering a food web.
Gotcha.
So I love the visuals in that video. I'm not a big fan of the overly dramatic Americanization of the bad neighborhood to hang out in. Twice the length of a man. There's a whole bunch of language choice there. Looking for a feast that you could do. However, if you do want to terrify some people as a way of teaching them, I'll let you know that this sand striker comes from a lineage that has been around for a long period of time. And in fact, there have been fossils found in Ontario near Moosonee on the lowlands of the James and Hudson Bay that have been worked on at the Royal Ontario Museum in Toronto that are likely 2 meters long. And when the paleontologists at ROM named it, they named this sand striker, 2 meter long sand striker, after Alec Webster, a giant of a bass player. There. And I had to look up who Alex Webster was. So were giant sand strikers a thing of the past? Turns out, no. This is one that was found in Japan that's length goes all the way across a road, nearly 3 meters long. And then there are people that have been kind of going through collection records around the world through time to say, yeah, there have been the reports of some very, very large sand strikers. Now, sand strikers, we're going to do a bit of an experiment here. Sand strikers, the phrase from that Smithsonian video, it's a bad neighborhood to hang out in, unless you've got friends. So let's tell a story here about an Indonesian fish, Scalopsis affinis, that schools they share a habitat with sand striker worms. And in the late 2010s, some were observed directing sharp jets of water, like the diagrams here at the bottom, towards the worm. So one of the fish identifies the worm, then they all come in and basically start spitting at the worm. Then they do it not just as solo, but then a group of them joins and starts spinning. And the mobbing causes the bristleworm to retract. What does it look like? Kind of like this. So the sand striker is right in here. So you can see the fish schooling around the predator and blowing and calling attention to it, not letting members of the family, presuming that the school contains relatives, not letting them fall victim to this cryptic predator, but calling attention by blowing on it. That's kind of amazing. So the hypothesis is that there might be social benefits for the conspecifics. So there's a risk, one would presume. But let's talk about that. Find your annotation tools. And if there are social benefits to conspecific fish spitting at this predator, draw what you would expect this relationship to look like, where there's 3 categories across the bottom. The first fish that finds the worm, the fish that finds the worm, the first participant that joins, and then all subsequent participants, and the number. What kind of investment do you think? So think about the y-axis as an investment. Do you think there's the same, A positive investment change or a negative change? This is where to get things wrong. Show me your hypothesis on the screen. Awesome. Thank you. Awesome. Couple more seconds, and I see the question, Zara. Do they swallow or chew? So it's swallowing. Okay. Thank you. This is amazing. Let me pause that. So most of the hypotheses are some kind of either a direct positive relationship or kind of a with a curve. Let me clear those, and I'll show you kind of categorically. You drew some continuous things. That's fine. Categorically, it actually goes the other way. And let's unpack why that is. And so it's a large investment so the investment from the first fish that finds it is the biggest. The next fish that finds it benefits and contributes, but not as much. And then there's all of the rest of the family that didn't find it, that still benefits, but doesn't have to invest as much. So this is an expression of the social. So subsequent conspecifics don't have to risk. So this is the social benefit for the risk that the very first individual holds onto themselves. So if everybody's related, this is still beneficial to you, because even if you get eaten here, the rest, your relatives in your school, will benefit. Now, the strange thing about this study is that the cases with predation are excluded. I would really love to see the data to see, because my guess is that it would go up this way, the other way, that the fish that finds are generally more likely to be eaten. So, now, lots of you, I've been saying sand striker all along. I'm going to encourage you to go to some of these links, scan the QR code later on, because there's been a real movement to not call this the bobbit worm. This is what it was called when I was in school, because when I was in school, at some point in the early 90s, there was a horrendous case of Lorena, her name is Gallo, not Bobbitt, and her husband, John Bobbitt. John Bobbitt is a domestic abuser, a serial rapist, a horrible person, and she fought back. She fought back in a very famous way. And so we call this a sand striker because if people had called it like the Loreno Gallo, I would say, okay, that's maybe different. But by focusing on the abuser, don't name the wonderful animal after a horrible person. You can read more about this here later on, or you can watch a Jordan Peele kind of produced documentary, I think, on Amazon, I think it is. We've talked so much about corals. This is one kind of nod to corals and the intricate relationships. There is a polychaete worm off the Danish and English coasts in the Northeast Atlantic that lives in holes provided for it, or that it burrows into coral. And this polychaete, the same genus as the sand striker worm, strengthens the entire network of coral by thickening and connecting coral branches. So in creating the environment of the coral reef, there's the coral, there's the dinoflagellate, there's the apicomplexan, so already 3 different phyla, and now we've got the annelid, so 4 different phyla. This is just a network of incredible relationships. Caramel says, look down, we're almost done. All right. Here's the movement. These are the parapods I was talking about earlier. So this is kind of a slow crawly one. But then this is Tomopteris, the sports car of the polychaete worms. They've passed some of these. They're just gorgeous fast. And there's that coordinated action. And they look like, they look almost like centipedes, kind of in your bathroom in the middle of the night. The Cutagera scuda, the European one that's in all of your houses. Now, before we leave, I want to talk about circulation, because they have a huge amount of it. You can see this is a parapod, and looking at all of the vesicles that are carrying the hemal system and the hemal fluid around, because I also want to make sure that we talk about the fact that polychaetes, annelids, have the most diverse set of respiratory pigments that are known amongst animals. They have, actually this is wrong now, they have 4 of 4. So they have hemoglobin, they have chloroquine, they have hemirythrin. I'll show you the other in a second. And they have them in the coelomic fluid. And so hemithyrin, chloroquine, hemocyanin, and hemoglobin. So the greatest diversity of oxygen-carrying respiratory proteins exist in the annelids. And if you look at some of these names here, the phylogenetic distribution of these respiratory proteins is kind of amazing. We'll talk about that in days to come. Last couple of things I want to talk about. So we've talked about nephridia, we've talked about protonephridia. This is a simple metanephridium. So this is what will be replicated along the length, the metameric segments. So we have the outside where we've got, and then the inside. So the nephrostome that's collecting from the coelomic fluid, that concentrating it through a nephridial tubule and then excreting it to the outside through the nephritophore. And this kind of like bent angle tube, like you're plumbing underneath the sink, that increases the surface area and allows there to be the reabsorption of otherwise beneficial molecules that might so water would be one, but all there other molecules that are in there that you can still benefit from. This is a chance to excrete the things, the byproducts, principally like ammonia, that are toxic, but to still, so they'll be excreted out here to the outside of the organism, but this gives you a chance to retain things, molecules that you can still need to grow. So their circulatory system is kind of and so in the last, we've got this little simple bag, and we'll ground ourselves here and say, there it is. There's the nephrostome that's joining in between those metameric segments. Here's that bent dog's leg of a long absorptive, and there's the nephridium that leads to the nephritophore that is then, you can see, replicated along those metameric segments. So the nephrostome is picking up coelomic fluid that filters along the length of that dog's leg, and then out through the nephritophore into the exterior. So instead of there being, like in the protonephridia, a flagellum that's driving this, it's the circulatory system that's providing the pressure. The last kind of one of the last amazing things for today about the annelids is that they have a new kind of larva. This is the Tom Selleck of larva, because the trochophore is this planktonic larva that has a mustache, these bands of locomotory cilia. So yeah, that's Tom Selleck is a famous mustache of the 70s and 80s. So you get my very dated thing. All right. And then this is tracking kind of where the tilia, there's the growth zone, so that's still here in the larval stage. But these, whether or not the animal is active as an adult, these are active parts, active swimming parts of the holoplankton. And these larval stages can actually form a great big part of an economy, a food economy. So you can go and look at odoo worms. Odoo worms that not just reproduce with those larval stages, but actually break off. And so these epitoques, they become a thing that people will want to eat. And you can, there's a video about Palolo worms in Solomon Islands. So this is where we have been. A lot of annelid diversity, and there's more coming. We're going to spend a lot of next week on this before we switch to the mollusks. Okay, so what are we doing next week? We'll finish with annelids, start with mollusks. Your jobs for the weekends are to prepare for your next lab. Make sure to complete your sponge and cnidarian lab quiz. Your week 5 quiz will be due next Tuesday, and by the end of next week, your species naming assignment is due. So thank you for your patience today. This is exciting stuff. I hope you share some of that enthusiasm. Over the weekend, get out, do some good things for you. Take care of your brain. So take care of you. Take care of others around you. I'm going to hang around for a second if you've got questions, but I'm going to stop recording and say, have a great weekend.